A lot of you've done this before. Those of you that haven't, it's going to be a little bit hard to understand, but you just raise your hand. This isn't some of you are more molecular than others. There's no shame in not knowing what's going on. I'm going to make sure you understand it. But we'll do just a little bit of that, and then we'll get into evolution and diversity just briefly. I won't go through that whole second lecture I posted. I'll just go through it briefly. I'll get it started, and then we'll finish that up Thursday. Important things you might have noticed, I did post a second reading. And please just try to have that read by Thursday. We'll talk about it at the end of lecture then. It's a science paper. It's an actual experiment. The other one I gave you guys was an experiment. So I have a couple of classes with a lot of reading. It's hard to keep it all straight in there. So yes, it's an experiment. And it was a little bit of a controversial experiment in terms of the quality of it and the conclusions they made. So if you don't buy 100% of it, then that's valid. And it would be fun to talk about why you don't buy it. All right. Questions? Can I go over anything we've gone over in the last week or so? Is anyone confused on a concept, wanting to hear something again? Okay, all right. Yeah, you can obviously come see me after class or any time if you come up on something. Okay, so what I'm going to talk about today, just briefly, is the recombinant DNA and the genetics and the way we look at microbes in the lab from a molecular standpoint. So a lot of you have done a fair amount of molecular work, but you probably haven't necessarily done it with respect to microbial populations, bacterial populations, or bacterial communities, which is what I'm going to end with. And I actually do my work in my lab on bacterial communities and fungal communities as well. So I introduced you to the idea of a restriction enzyme a week or two ago. And I was a little stumped that none of you had heard of it. So can y'all give me some more confidence? Someone tell me what I described a restriction enzyme as. Okay, we'll get to that today. I would spend some time learning that before next Thursday, but we will define it again today. All right, PCR. Tell me you guys know what PCR is. What does PCR stand for? Okay, good. Okay, we're starting off on the right foot. Okay, everyone knows the polymerase chain reaction. So let's just step through it one more time for those of you that don't remember the exact details of it. And what PCR does is it takes a known fragment of DNA and it amplifies it into a billion copies. All right? So what you do is you have certain chemistry in your PCR reaction. You have an enzyme, a DNA polymerase enzyme in there. You have a bunch of DNTPs. What do I mean by DNTPs? What's a DNTP? What's an example of one DNTP? You have one of four ways of getting this right. It's a hint. Say it loud. A, G, C, T. Your nucleotides, okay? So you need all four of those nucleotides in mass quantities if you're going to extend out a length of DNA. Those are all the little pieces that make a strand of DNA. These guys, A, G, C, T, nucleotides, adenine, guanine, cytosine, etc. So you need those in there. You need some salts and some buffers, etc. And then you can affect the elongation and the annealing of this thing by changing the temperature. So what you can do is you have a strand of DNA that you want to replicate, a known piece you want to replicate. All right? And I missed something important. What did I miss in my description of what you need in this mix besides salts, DNTPs, and fat? That's one of the most important things. Primers. Beautiful. Good. You guys are doing great. You need a primer. And the reason you need that primer is you want that primer to find the gene that you are interested in. Okay? That primer sits down on the DNA piece that you care about, that you want amplified. Otherwise, you're just amplifying random DNA, which actually is a method sometimes used, but you don't necessarily want to do that. So you have this chunk of DNA that you care about, that you want to amplify, and you have a primer for that region. So the first thing you're going to do is you're going to melt that DNA, and you melt it apart. And then your primer, and then you heat it up really hot, 95 degrees Celsius or so, you heat it up really hot. And then you cool it down a little bit, and your primer attaches. And then you elevate the temperature a little bit so it'll elongate. And when it elongates, that primer's attaching, it's found just the right spot, and then the nucleotides are going to lay down in a row, basically anti-parallel to your single strand there. And you continue to cycle this over and over and over. Melting it, separating it out, annealing a primer, elongating. So you're melting, making two strands into two a double strand into 2 single strands, doubling up those 2 strands, and melting it again and again. So it's exponential growth of what was once a single strand, 1 strand or whatever, how many you had in there, turning into thousands, hundreds of thousands, millions of strands. All right, and you can do this through time through various cycles. This cartoon gives you 3 cycles. One of my favorite reactions, I use 35 cycles. But it depends on what you're up to, however many cycles you want to do. But if it's doubling in every cycle, then you get to some pretty big numbers and you're amplifying BCA. Amplifying. All right, so PCR you guys are all pretty comfortable with. So in that reaction mix, first thing up there, primers, good, you knew that. Target DNA. You need a DNA polymerase that can handle hot temperatures. Okay? So you need a DNA polymerase that's TAC. It's one that a lot of us use that can handle extremely hot temperatures. And those DNTPs, the adenine, guanine, cytosine, and thymidine. All right. So once you've done your PCR reaction, how do you know if you were successful? How do you know if you took 1 strand of DNA and made millions? Runs gel. All right. And the way a gel works is there's 2 main kinds of gel. There's a polyacrylamide gel that you probably don't deal with very much. It's much more sophisticated for proteins and higher level assays. Most of you probably dealt with an agarose gel. And agarose is just a sugar, and it makes this gelatin. I don't even know how to talk about it. Those of you that have done it know what I'm talking about. Anyway, it's a matrix, okay? And you're going to pull your DNA through that matrix based on its charge. So what you do is you set up, you have a system that will allow you to pass this little jelly gel thing. Can't think of a better way to say it. And you'll put a comb in there so it'll have wells. And you take each of your PCR samples and you inject it into one of these wells. Okay? And then you run a current through it. All right? You run a current through it where you have a positive charge at the end and a negative charge there. DNA is negatively charged. DNA is going to want to run to that positive charge. So any DNA that I amplified in my known little fragment from that PCR, any DNA that I amplified there is going to run through that gel matrix, that agarose matrix. You can make it more or less concentrated as you need. But what it's going to do is it's also, which is going to become very important in a couple of methods we're going to talk about in a minute, it separates them out based on size. Okay? So everything's getting pulled according to charge, but extremely large fragments of DNA are going to be slower to move through that matrix than really short fragments of DNA. So if you have a band of DNA that's farther along the gel, it's likely a shorter length than one that's not as far through the gel. It's likely a smaller fragment of DNA as opposed to a larger fragment of DNA. And you always run a ladder with this, a size marker, to be able to tell, a known size marker. You buy it from a company. All right, and a typical gel, this is actually a pretty nice looking gel. I wish mine always looked that good. So what it will do is they have different fragments here, and there's their ladder running out, and they know the different base sizes of that ladder according to the manufacturer's information. All right, so let's define restriction enzymes one more time. A restriction enzyme is something that's very naturally occurring in cells, but it's been used in molecular biology as a really, really valuable tool. If you open up a catalog and try to buy a restriction enzyme, there's probably hundreds of them in that catalog. But what it does is it's just an enzyme that cuts DNA at a particular sequence. All right, so let's look at this. In this particular case, we have a restriction enzyme that wants to cut between G and A and that spot. It also will know some of the flanking nucleotides. I'll show you that in just a second. So every time it comes across this GAATTC, it makes a cut. All right? And in making that cut there, it breaks it up into different fragments. Okay? And so it recognizes that spot. That spot could be anywhere on a strand of DNA, and it's going to cut it. So going back to some things we learned a few weeks ago, why do you think cells naturally make restriction enzymes? And in the next few slides, I'll tell you why we use them. But why do you think cells would naturally want to make a restriction enzyme? Think about transformation. Even conjugation. Yeah.
To incorporate DNA.
Yeah.
Spots for the coming.
Sure, to digest it in, to make a spot. Sure, if you want to be a recombinant, absolutely. There's a lot of different ways to answer this correctly. Recombination, so what I think what Mike is saying is that you can incorporate that DNA into your own chromosome and now have new information in your chromosome, which is evolutionarily adaptive. But also, just from the simple sense of nucleic acids are valuable energy resources, and sometimes cells will take in foreign DNA. Sometimes they'll incorporate it and become a recombinant. Sometimes they'll just digest it and eat it. All right? Sometimes they don't necessarily want to take it in, and they'll digest it and scavenge those nucleotides and that nitrogen and phosphorus as energy sources. So there are a lot of reasons for them to make it, and there's thousands of them out there. And in molecular biology, we exploit them because sometimes we want to cut and paste DNA. Okay, so as I said, there's thousands of them out here. Here are about 10 of them. All right, and so let's go to the first one, ALU1. I've never used that one, but you'll notice it recognizes a 4-nucleotide sequence, AGCT, and it cuts between the C and the G. All right, so that's that one. BAMH1 notices a 6-nucleotide sequence. And it cuts, in this case, between these two Gs. So anytime you have that 4-nucleotide sequence, it's going to make a cut between those two Gs. All right, so let's just discuss fundamentally, you do not need to memorize all these. You don't even need to memorize one of them. But let's do understand, fundamentally, what's going to be the difference in an enzyme there? What's going to be the difference in these two enzymes in terms of their functionality in cutting DNA? Well, what's the difference between one that recognizes 4 nucleotides versus one that recognizes 6?
The top one's cutting straight through, and the other one's leaving the sticky end.
Okay, that's part of it. That's not the difference between the 4 and the 6, but that is important. I was going to go there too. He noticed, and he's absolutely right, that this one cuts straight through and makes a very blunt cut. Whereas BAMH1 leaves really nice, long, overlapping sticky ends. So if I do want to cut and paste and clone, which I'm going to show you in just a minute, that will allow you to do that really nicely. Okay, so he's dead on there. But someone tell me, if I'm randomly going out and cutting DNA, and if I'm looking to recognize a 4-nucleotide sequence versus a 6-nucleotide sequence, how is that going to manifest differently? Someone besides Mike. Yeah.
The first one costs more because probability of having 4 nucleotides in sequence is more than 6.
Thank you. Thank you. He's absolutely right. Thank you. So if you have what we call a 4-cutter, you're going to make a whole lot more cuts, all right, than if you have a 6-cutter. The odds of coming across a nucleotide sequence that's AGCTA is a whole lot more likely than the odds of coming across one that's GGA, whatever. All right, and you can use this to engineer your own molecular biology. Restriction enzymes, doesn't matter what you're doing in a molecular lab. Every molecular lab in that building over there probably has half a dozen restriction enzymes sitting in their freezer. You use them constantly. All right. So that leads really nicely into cloning. So we've all heard of cloning in the fun and interesting sense, in the controversial biological ethical sense. But in microbiology, and certainly the kind of community microbiology that I do, we use it constantly. And not just in community stuff. In any instance where you need to look at and grow a whole lot of a certain gene, cloning is a beautiful way to do it. All right? And what you're doing is cloning is you're taking a known piece of DNA, very likely that you've acquired through PCR, okay? And you want to grow it up into a cell. So let's say you want to manipulate or engineer a cell to turn on a certain protein. Okay, have any of you heard of GFP? Some of you, a few nods. GFP is cool. You can use it as a marker. GFP makes cells grow green. They can make GFP mice. They can do all this neat stuff. Whatever, you get the point. You can take a gene in question and then clone it into a cell and make that cell express that gene. All right, and then I use cloning a little bit differently than I'm not too worried about you guys getting today. Okay, so you have a cell, you have a known piece of DNA. You cut up that DNA with a restriction enzyme. Okay? You cut up your piece of DNA with a restriction enzyme, and then you anneal it into a vector. So what does the word vector mean to you? Not even in this context. What does the word vector mean to you? An insect? Why an insect?
It's like transport little bacteria or viruses.
Okay. And what was the operative word she said there?
Transport.
Good. Cool. Okay. Some of you are with me. Awesome. So transport was the right word she said there. So what you do is you take your chopped up pieces of DNA and you anneal them into a vector. In this case, it's a plasmid. Okay. Vectors can also be things like phage can serve as a vector, if that makes any sense. You all know what transduction is now, right? Okay. And you can take that vector, here's your plasmid vector, here's your little piece of DNA that you want to move. You stick it into that plasmid, and you transform, now you all know what transformation is, you transform that plasmid into a cell. We call that cell a competent cell, and I can buy them. I can open up a catalog and buy cells that want to be readily transformed. All right? And then you can grow 1 billion copies of that cell, and then you effectively created a little machine to express your gene, to grow your gene in question. All right. Okay, so let's talk just quickly about vectors. Again, most of the time it's a plasmid. Plasmids are the easiest to work with. Okay. Replicates independently of the chromosome. Can do that. It can get easily moved from cell to cell. Okay, and there's a lot of other methods of transformation and moving a vector as well. Bacteriophage is another method. But the method you're going to see most frequently in any molecular lab you go into, almost any, is going to be likely a plasmid. And you can buy them. You can order them from a company. Okay, these plasmids that you order from the company do lots of really neat things for you. So, for instance, this is a plasmid I could easily order. It has an origin of replication. It has a LAC-Z gene. Okay? And does anyone remember what LAC-Z does? Okay, that's part of the lac operon. It helps the cell degrade lactose. Okay, so that's important. And then AMP-R. So you guys got asked on the last test, where does antibiotic resistance normally go? What kind of thing?
R
You said
plasmid.
the R plasmid. Okay, this is basically been engineered into being an R plasmid. So what this cell does is if it has this plasmid in its cytoplasm, then it's going to be resistant to the antibiotic ampicillin. Again, very handy for manipulating this thing in the lab. All right, and you can have them more complicated than that. But then what they'll have is at various locations around this plasmid, they will have restriction sites engineered in. So remember, the restriction enzymes recognize particular sequences of DNA and make a cut. So if I have engineered, especially right in the middle of that LAC gene, which you'll see why that matters in just a second if it's not automatically jumping into your head. If I have a restriction enzyme, a restriction site in there, I can very easily cut open that plasmid to insert my little piece of known DNA. And if I happen to have cut my known DNA and this plasmid with the same restriction enzyme, anyone following me? Cut it with the same restriction enzyme, and it automatically makes the sticky ends so that this thing will stick together. Okay, let's look at that a little bit more deeply. Okay, so this is how you actually insert your little chunk of DNA into the vector, and then how you screen for it, and why all these things matter. Okay, so you have a plasmid vector, which has a LAC-Z gene on it, and it also has an AMP resistance gene on it. And it has this restriction site. It happens to be EcoR1. Just remember that name. It's just one of the restriction enzymes. It has the site for that restriction enzyme right in the middle of its LAC-Z gene. So if I go and I take this plasmid and I put it in a soup and turn up the temperature and expose it to that enzyme, it's going to cut at that site with the enzyme in there at the right temperature for the right amount of time in the right soup. Okay? But if I also take my known DNA that I would like to clone into a cell, and I cut it with the exact same restriction enzyme, then it's going to make complementary sticky ends. I hope this makes sense. It's going to make complementary sticky ends where you have CTTAA, CTTAA on your complementary fragment. So it's going to very easily just stick right in to my plasmid vector. Okay? So that's a handy thing. Now I have my clone, my gene in question that I want to clone. I have it in a vector. I have it in a plasmid. And I need to get that plasmid now into a cell. The cell I want to get into, competent cell. It's just usually a generic version of E. coli that they've engineered somehow that we all order through the mail. So you put your plasmid in with your cell in broth, cook it for some period of time. That cell gets transformed. One of the many reasons I've taught you guys about lateral gene transfer and transformation. Okay, that cell gets transformed, it takes up that plasmid. So now we can grow that cell on media, on a petri dish, just like you guys have been doing in lab. But we're going to do a couple of different things for quality control and to make sure we got it right. One, I'm going to put ampicillin in my media. And why am I going to do that? As obvious as it sounds, just I want to hear you guys, I want you guys to wake up. Why am I putting ampicillin in my media?
Because it make sure that it's resistant.
Make sure that I'm getting the cells that are resistant. If they're not resistant, okay, why would they not be resistant?
Because they're not the cell that you wanted in another one.
They didn't get the vector. Okay, cool. They didn't get the vector. Good. So I put the ampicillin in the media to make sure that they can only grow the cells that got that vector that has the amp resistance. All right. I also have lactose in the media. Okay. So that cells that can degrade lactose will grow a certain color. I always forget on the slide if it's blue or white. Don't worry about that. Cells that can degrade lactose will grow a certain color. Cells that cannot degrade lactose will not grow a certain color. So not only do I care, did you get my vector at all with this amp-resistant gene, but I also care, did you get the vector that got cut open at the lac gene and inserted my gene into it? So by inserting my gene into it, by cloning my gene into this vector, I disrupted that lac gene. So any cell that's got my gene in it, in that vector, cannot grow on lactose. So it will grow a particular color. And I can pluck that cell off and then go on and do sequencing or manipulate it or engineer it or whatever I want to do with it. All right, so there's a lot of different screening techniques for vectors. And this is one of the most fundamental, simple ways of cloning. So we think about fun, interesting cloning like growing a sheep and injecting it in. In a microbiological sense, it's really just about using the cells themselves and cloning genes. Okay, so one of the things that can come out of this is you can, I just spent the last 5 minutes counting you guys that you can clone a single gene. You can clone a whole suite of genes. Clone many genes, all right? And you can create an entire library of this. And then you can go sequence those genes, all right? So if I have an entire group of genes that I'm interested in, or an entire group of bacteria that I'm interested in, then I can use those exact same methods, cut them into vectors, and I don't even necessarily know that they're all the same. I don't know that they're different, and create what I call as a library. And then you can go through and sequence each one of those things and then find out what you've got. Those libraries can be screened for genes of interest or individuals of interest, depending on what you're doing. Okay, so the way I might look at different genes of interest is, again, there you have your vector, you have your plasmid up there with an amp-resistant gene. You can cut up your foreign DNA into different pieces, and it gives you multiple different fragments. Okay? And then you end up with basically vectors that have either a green fragment or a red fragment. We don't necessarily know. They get transformed into a competent cell, and you grow them up on media, and then you can manipulate them and use them, and then you can sequence them and know that the red fragment was different than the blue fragment. Don't worry about this too much. Don't let this hang you up. Okay, so let's get into sequencing real quick. So the next step from many cloning steps is sequencing. In sequencing, you're really looking at genomics in a sense, at a micro scale, and then you can do metagenomics, which is a bigger version of sequencing many, many different things. But it's the study of the molecular organization of a genome, basically. The genes themselves, the sequence themselves, and then you can figure out what they code for. All right. So the Sanger sequencing method is the classic old school method of sequencing genes. There's a whole lot more modern, faster, sexier ways of doing this now, but this works beautifully, and it's still the classic original way that people sequence genes. Anyone here done any sequencing? Okay. A little bit, maybe? Okay. Hopefully you've been introduced to this idea, though, in your cell molecular classes. All right, so I have that isolated piece of DNA fragment. Maybe I cloned it, maybe I PCR'd it. There's a lot of different ways to skin that cat. All right, so I have my piece of DNA that I care about, some sequence that I care about that I need to find out that sequence. I don't know that sequence, I need to find that out. All right? So the first thing you do in a sequencing reaction is obviously you denature it, you melt it. You heat it up really hot, melt it so that it's single-stranded. All right? And if I want to know the entire length of this thing, I have to have some kind of a primer for that gene. So if I'm looking at that gene and the difference between, say, species A and species B, the sequence of that gene, I know the gene in question, and I know its primer. I don't necessarily know that whole sequence. So you add a primer to your mix. Okay, and this is looking a little bit like PCR, and it's going to act a whole lot like PCR, but there's some really, really important differences. When you add your nucleotides, your GC and your A and your T, they're not just normal nucleotides. Okay? They are double deoxynucleotides, DD nucleotides. All right, so they're a little bit different, just a little bit different. So that as your sequencing reaction extends out, every time it hits a double deoxynucleotide, it's going to stop the reaction. All right, so the way this works is that you have basically 4 different sequencing reactions. And this is the part where people get hung up. Once you get this, it'll make sense. You have a G sequencing reaction, a C one, an A one, and a T one. And in the G sequencing reaction, I have all the nucleotides present, but in that case, the G's are double deoxy. So that any time a reaction gets stopped in the G tube, we know it ended on a G. Okay? Likewise, in the CA and T tube, C tube has all the nucleotides present, but the C nucleotides are double deoxy. So every time you have a fragment length that stops there, it's a C. It ended in a C. So you're making a million little copies or short fragments that got cut too short. And in this tube, we know they're all G at the end. In this one, we know that they all end in C, end in A, and end in T. You can see that here as it extends out. In this case, it got terminated right there with an A. In this case, it terminated with a G at the next spot, a C at the next spot, and another C at the next spot. So after you've done these reactions, you run it out on a gel. And if you remember something really important I told you earlier about running out on a gel, longer fragments left up at the top. Shorter fragments move through faster. They all move according to their electrical charge, but the shorter fragments move a whole lot faster than the longer ones. So if I have all these fragments in there, this one right here that's just CACTTA, this little piece, when it melts, is obviously going to move way faster through the gel than this long fragment will. And so then you run out that gel, and you can just easily go through and mark. You run each of these 4 tubes in each of 4 wells, a G well, a C well, an A well, and a T well. And then each fragment that falls through, you know it ends in that nucleotide. All right? So you can very quickly calculate the sequence, add up, calculate's the wrong word, add up, determine, etc. The sequence of your fragment by looking at this. So the smallest, shortest fragment was an A, and then a G, and then a C, and then a C, and then a G, A, T, C, C. So now I know the sequence of my fragment is A, G, C, C, G, A, T, C, C. Okay? Because the shortest piece ended in an A and went all the way to the bottom, and the longest piece ended in a C and stayed up at the top because it was a really long piece relative to the others. That Sanger sequencing is the classic sequencing method. And anything that's, some of the newer methods, pyrosequencing, things like that, have all evolved out of this principle.